can see what slide we're on here. Oh no. Amateur hour over here. Oh, I was trying to get it so they could see the. There we go. So they could see the screen, see where we're at. Okay, so this is week 4. It's hard to believe. It's just the semester is going by quickly. It's actually a little bit warmer outside. COVID cases are down per day, you know, so things are looking up, aren't they? And last week we managed to have a snow day. I thought we were going to have one before too long. And so luckily I finished proteins mostly on Tuesday. And if anybody has any questions, I'll entertain them now. I don't have a lot more to say about, or anything more to say about them really. Well, actually I do. I can always come up with something to say about proteins. For those of you that are very curious about like how things work at the cellular level, and how science moves forward sometimes, there's a really interesting book out there called Quantum Biology. And as soon as I said that, I of course forget the author. I'll send out an email if anybody's interested. But the way that we teach how enzymes work, and of course most enzymes are proteins, and you remember enzymes, they speed up chemical reactions, right? For both breaking things down, which is a catabolic reaction, or building things up, which is an anabolic reaction. Your protein enzymes, they facilitate these reactions. They make them go faster. And interestingly, for the better part of 100 years or longer that we've known about these things, it's been thought that enzymes use they have an active site, and that active site takes your substrate, which you're going to react with, and moves it to the correct position to help make those bonds easier to break and facilitate this chemical reaction. So the reason why the paper on your desk, which is made out of cellulose, doesn't spontaneously combust, or why you don't spontaneously combust, is because there's this activation energy. So this is the energy of your reactant. So if you're taking something like a carbohydrate, gasoline, a lipid, it stores a lot of potential energy here. And then this is your activation energy. So this is like when you start a fire, or you have a gas stove and you need that little spark to get the gas ignited. This is your activation energy that breaks the bonds. And then this is the amount of energy that is released in your reaction. Are we good? Okay, yeah, you'll learn more about this with Dr. Adama. But interestingly, it's always been assumed that, here you go, guys. It's always been assumed that an enzyme lowers this activation energy so the reaction can go forward. And we've always thought that this is just kind of a mechanical, classical physics problem. And it turns out that the enzymes, protein enzymes, there might be more to this story. And the more to that story is, rather than lowering the activation energy, that enzymes might use this really weird thing called quantum tunneling. And I won't test you on this at all, because I'm not even sure exactly how it works. The world of quantum physics, that's the physics at the scale of atoms and subatomic particles, is really weird. And it's always been thought that those really weird things at the quantum level don't matter so much for us, that we're too big for that. But imagine this is a hill you have to go over. This is the energy you need to add. It's always been thought that enzymes lower it. It's like lowering the hill. Now we think that a lot of enzymes might use this really odd thing, or not odd, that's how the universe works at the scale of atoms, called quantum tunneling, where the electrons go through the energy barrier. You may not actually lower the energy barrier. You may not actually go over the mountain. You go through the mountain. And for you and I, sitting here at the base of the Sandias, thinking about going through the mountain seems almost absurd. But that is how quantum tunneling works. In fact, this is part of how bonds are formed in the first place. And like I said, it's always been thought that this was that a molecule like an enzyme was too big for this to occur. And there's now very compelling evidence that enzymes actually, it's more than just lowering activation energy. they're helping the electrons go through the energy barrier, not lowering the energy barrier. And that's actually quite a significant find. So if I teach this class in 5 or 10 years, that's what we might be teaching with enzymes rather than lowering energy barriers. Just to let you know, science is always changing. And that's where it might be in 5 years from now. Or 10 years, who knows? Any questions? Okay. Well, don't worry, I won't test you on that. But it is kind of fun to think about that our knowledge of something we think pretty fundamental in biology is still changing. Okay, any questions? All right. So there'll be a third homework assignment. I'll get it posted tomorrow. It'll be due on Sunday. Also, by Thursday, you'll see test 1 will be available. It will be online. I'm not going to put a time limit on it other than the due date, okay? So I am expecting you guys to look the answers up and research them. I expect that. So I expect really good grades, basically. Be thorough, okay? All right. Any questions about the test? Multiple choice, there'll be some multiple choice, there'll be a few short answers and a couple essays. And then the homework will still be the multiple choice, like the same format. And wait, that won't be this week. I'm sorry, that'll be next week. Sorry. I almost always give my test on the 4th week, except in here. And that's an artifact of other people that teach the class. Okay, let's talk today about nucleic acids. So, like the chapter on proteins, I could blow through this pretty quickly, but I'm not going to. I might get done a little bit early today, but that's okay, because what I'm going to do today is basically talk about the nuts and bolts of DNA. And if you're sitting on your cell phones while I'm trying to talk, that's kind of not the nicest thing to do. It's kind of rude. Okay, so we're going to talk about the nuts and bolts of nucleic acids today. And then on Thursday, we're going to talk about evolution, the origins of life from, well, we'll just talk about origins of life. And what I want you guys to do is evaluate this RNA world hypothesis before Thursday. Go Google it and see what comes up. See what people are saying about the RNA world hypothesis that the book talks about. And think about like the book's definition of life. Think about the definition or the way I think about life. Think about the way, or go look up NASA's definition of life. They're 3 different things. And then start thinking about this RNA world hypothesis and what the book what the book is saying. And let's have a discussion on Thursday of whether or not what you agree with that or what. That to me is very fascinating. So look up the RNA, just Google RNA world hypothesis before Thursday. It's kind of interesting to think about. But before I do that, let's talk about the nuts and bolts of nucleic acids. So nucleic acids include, of course, both DNA and RNA. Okay, so we're just going to do the structure of nucleic acids of an actual individual nucleotide today. So we're going to look and see what a nucleotide is. Then I'm going to talk about the structure of DNA and the function of DNA, which is pretty straightforward. And then I'm going to talk about the structure of RNA. And I'm just going to touch a little bit on their different functions and the versatility of RNA, which is pretty wild. And then we'll be able to compare the structure of DNA to RNA. We're going to compare and contrast the 2 structures because that's very important. Even though they're both made up of nucleotides, they do different things. They do similar things, and then RNA can do different things. And why is RNA so versatile compared to DNA? And then, of course, that comes down to the structure, starting with the difference in the nucleotides. And then let's evaluate the RNA world hypothesis. That's what we're going to talk about, origins of life, on Thursday. And I'm also going to talk about some stuff that's not in the book about the origins of our genetic code, which to me is absolutely fascinating. It's not random. Let's put it that way. Okay. Let's get to the nuts and bolts here. Nucleic acids, which include both DNA and RNA, have some similarities with proteins. And that makes sense because, your nucleic acid, your DNA codes for proteins. The information stored in DNA codes for a protein. And what they are both polymers. Polymers mean that they're a large complex molecule made up of individual building blocks. The building blocks are the nucleotides. Okay? And there are lots of different types of nucleotides. DNA and RNA both use 4 nucleotides. I'm going to go over their structure here in a second. And there's one different type of nucleotide, or they each have a unique nucleotide. DNA, of course, uses thymine, and RNA uses uracil. I'm sure you've heard this, but we'll talk about that in a second. Well, these nucleotides, they start with a sugar base, a 5-carbon sugar that looks something like this. This is called a ribose sugar. I'm going to go ahead and draw a phosphate group on there as well. Now I've got a phosphose sugar. Normally there would just be a hydroxyl group here, but I'm going to go ahead and add this phosphate group on here because that's actually important. But this is a ribose sugar. It's got 5 carbons. There's a carbon right there. There's a carbon right there. So here's a carbon. There's a carbon. And we can number these carbons 1 through 5. So we would go 1, 2, 3, 4, and 5. Okay. Now, in addition to these 5 carbons, a nucleotide also has a nitrogenous base. And basically, remove that hydroxyl, and I'm just going to draw a pyrimidine, a simple one here. And that's our nitrogenous base. Now I have the basics of a nucleotide. And this also has carbon. Actually, most of them will have an N right there. You don't have to remember how to draw these. So if this ribose sugar has 5 carbons, 1, 2, 3, 4, 5, this nitrogenous base also has carbons that we would number as well, 1 through however many they have. Some of the nitrogenous bases have two rings or a double ring. This is a pyrimidine with a single ring. So to distinguish the carbons between your nitrogenous base right here and the carbons in the sugar, we give them a prime designation. That's why you see right here the little prime, and that's just to show to separate the sugar from to separate the carbon from the sugar from the nitrogenous base here. So 1 prime carbon, 2 prime, 3 prime, 4 prime, 5 prime. And of course, this is a ribose sugar. So you've heard of ribonucleic acid, right? Okay. Well, there's also deoxyribonucleic acid. And I'm sure you've heard of that, right? This DNA is deoxyribonucleic acid. So deoxy, D means without. So what that means is in a deoxyribonucleic acid, you're missing that hydroxyl group on the 2 prime carbon. That is one difference between RNA and DNA, is that in DNA, which stands for deoxyribonucleic acid, it's removed that oxygen, basically a hydroxyl group, from the 2 prime carbon of the ribose sugar. And that actually has quite a significance for this molecule. It's not trivial. And this is just showing it on the PowerPoint right here. Okay, any questions? Well, I would have one. How many of you have memorized that DNA has 1 less oxygen than RNA? Nobody's memorized that? I don't know. I memorized it when I was in high school biology, and then I memorized it in my freshman biology class. I could recite to you that RNA has a ribose sugar, and DNA has the deoxyribose sugar, and I could quote that pretty easily. I memorized it like you guys will. And honestly, it wasn't until much later that I started asking, well, why? What's the point? Why does it matter that ribose has an extra oxygen? It's actually pretty big. And this is really profound because it sets the stage for the difference in the functionality or the capabilities of RNA versus DNA. First of all, if I have an extra hydroxyl group here, there's several things going on. For one, this oxygen right here, it's of course electronegative. So it's going to hog some electrons to it. And it's going to pull electrons away from here and here. It's going to make that covalent bond have more potential energy. That means RNA is a little less stable. Just a little bit. But there's another big issue here. Covalent bonds are made by sharing electrons. And a chemical reaction comes down to making and breaking bonds, right? And it turns out that this oxygen here, because it loves electrons, can attack the bonds within a long strand of RNA, which we'll talk about here in a second. It can also attack the bonds of other molecules. That means that this molecule, the ribose sugar forming RNA, is more reactive. It can cause more chemical reactions to occur. It is less stable than DNA. And it doesn't last as long either. So it's more prone to doing other things. Whereas in DNA, by removing this hydroxyl group, all of a sudden, this molecule is a little bit more stable, less potential energy there. But you also don't have that hydroxyl group swinging around and attacking the bonds of other parts of the RNA or other molecules. So it's less reactive. Well, that makes sense, doesn't it? I mean, what stores all of the genetic information in our bodies? DNA, right? So if you're going to store information, you want it in a more stable molecule. So that's one of the reasons why life evolved DNA, to use DNA, was to store genetic information for every living thing on this planet. And to do it as stably as possible. Whereas RNA, you're going to find out, is a pretty wild molecule. Yes?
Is there a reason why it's on the 2' and not the 3'?
There is, actually. That's a very good question. And it comes to how we make DNA and RNA. Whenever we synthesize, we synthesize or polymerize those nucleotides, there's a certain way we do that, and I'll talk about that in a second. But good question. Okay, here's our nitrogenous bases. And the first thing that you'll realize is that there's two different classes. You've got the single ring, this is the pyrimidines, and then you get the double ring, which of course are the purines. And man, I tell you, it took me a while to figure out like how to remember which one was which. And one thing that I had to do is I drew them out like repeatedly, like once a day I draw them out when I was an undergraduate. And sure enough, I memorized them pretty quickly doing that. Now you guys having a take-home test, you don't have to sit there and memorize them. But the two ring structures actually do matter, and we'll talk about that in Chargaff's rule here in a second. But I always remembered the double ring structure is a larger molecule compared to the pyrimidines. And I always remember the inverseness of purines are larger than the pyrimidines. So it's like this inverse relationship. Does that make sense to anybody? Have I lost you? Yeah. Like I said, always remembering which one was pyrimines and pyrimidines. Like I said, the larger one is the purines, the smaller one is the pyrimidines, which are the bigger word. The other way I remembered that adenine and guanine were purines works for me, pure as gold. Purine equals adenine, which is as, gold, which is the guanine. And then when you have thymine, cytosine, and uracil, those are your pyrimidines, which are the single ring structure. And you notice in our nitrogenous bases, they have the nitrogen in here. So here's our hexagonal ring. There's our two nitrogens. There's our two nitrogens. There's our two nitrogens. And of course, you see uracil used in RNA and thymine used in DNA. How many of you have memorized RNA uses uracil, DNA uses thymine? Yeah, most of us have. Do you ever ask why? Yeah, I memorized it too, and I never asked why. I never thought to ask why. I just was like, oh, just a quirk of nature. There are quirks of nature, but there's also reasons why things are the way they are. If you noticed, uracil in the middle of your pyrimidines and thymine look really similar. You got your carbonyl groups right here, you got a nitrogen right here, and it looks like there's a difference this right here. So they are pretty similar. Then you look at cytosine, and you notice this is the big difference right there. And that double bond right there. Here's the difference. Thymine is a more stable molecule than uracil. It costs more to make than uracil. Uracil apparently is cheaper to make in terms of the amount of energy it costs to synthesize it. Okay. Well, there's always trade-offs, right? You ever bought a cheap car versus a nice, or something cheap versus nice? I have a bunch of cheap, shirts. They last a year and then they're done. You buy something nice that lasts for 2 years or longer. Thymine's more stable, but it takes more energy to make. Uracil is cheaper to make, but it's less stable. And actually what happens inside of our cells is that uracil tends to switch randomly to cytosine. Oh, that presents a problem, doesn't it? Because what would happen if your uracil and your DNA switched to cytosine, you would incur a mutation. Not only would you incur a mutation, your cell would have no way of proofing that. And that's something you guys will learn in genetics. You'll learn how the DNA is actually proofread. And there's different ways the DNA can proofread it. But if your uracil switches to cytosine, you basically, your cells can't detect that kind of change. I'm not sure exactly why, but it can't. So in DNA, they spend a little extra amount of energy to not only have a ribose sugar a deoxyribose sugar that's more stable, they also use thymine rather than uracil because thymine is more stable. Now RNA in your cells doesn't last very long. You're constantly making it, it's constantly being degraded. And as a result, if a random RNA molecule turns from uracil to cytosine, it's a one-off. It doesn't really affect the organism too much or the cell that much at all. It might render that particular mRNA useless or that whatever strand of RNA you made useless, but your cells can make more. Are we good? Yeah. You ever notice that viruses tend to mutate quickly? Like this RNA virus called the COVID? How many times have we heard about a new variant coming out? God, man. Wow, it's like every day there's a new variant. Well, there's a couple of reasons for that. One is there's a large reservoir of people that the virus is in. The more people that the virus is in, well, guess what? The more chances for mutation. But a lot of viruses mutate rather quickly because they use RNA as their genetic code rather than DNA. And because they're using RNA, they mutate more quickly. And if you mutate more quickly, you're going to come up with more variants. So there's real-world connections here. Okay, are we good? All right. I'm going to talk about that. Okay, here's another similarity between proteins and nucleic acids. A protein is a polymer made up of building blocks of amino acids. DNA and RNA, the nucleic acids, are both polymers made up of their building blocks, which we just talked about: adenine, thymine, guanine, and cytosine, and of course uracil if you're in RNA. Here's another similarity. They both have a primary structure that includes a sequence, a backbone of these monomers, of the building blocks. Mono just means single. So if I say a monomer, just think building block, right? An individual Lego piece. You build up a protein through what type of reaction? Remember? Condensation, dehydration. Yeah. You polymerize amino acids into a polypeptide. You can polymerize nucleotides into a nucleic acid. So when we talk about DNA synthesis, RNA synthesis, this is once again a polymerization. Polymerization just means I'm adding building blocks. I'm building my Lego Millennium Falcon, right? And of course, they do this through a condensation reaction. So you asked a question earlier. You said, why on the 2' carbon, not the 3' carbon? Well, it turns out that numbering system that I told you of 1', 2', 3', 4', 5' matters. Because if this is the individual building block, I've got to add additional nucleotides to this molecule to make, in this case, a DNA molecule. And over here, this is, of course, an RNA molecule. The way this works, here's your 3' carbon. Let me see if these guys can see me. Yeah, I think so. All right. These are the 3' carbon, 2' carbon. This is another nucleotide. An enzyme, in this case, RNA polymerase, poly just means many. This ASE means it's an enzyme. RNA tells you what it's making. Poly means many. So this is going to add RNA molecules to each other. So RNA polymerase lines these up and will attach a nucleotide to the 3' end here, on the 3' end. So this will come out, this hydrogen will come out. There's your water right here from this hydroxyl group right here. This hydrogen, now I will have O, carbon, oxygen, phosphorus. When this is all said and done, here it is. Carbon, oxygen, phosphorus. And the directionality here is 5' at the top, 3' at the bottom. And whenever you add nucleotides to each other, whether it's RNA polymerase making RNA or DNA polymerase, you're always going to add it to the 3' end. So I'm always going to add nucleotides to this side right here. And of course, everybody, if you go and look that up, you'll realize there are multiple types of DNA polymerases. They each do something slightly different. You'll learn about it in genetics. But basically, it's a broad class DNA polymerase. RNA polymerase will only add nucleotides to the 3' end. Any questions here? Okay. And of course, this is a condensation dehydration reaction. Condensation because you're getting water out of it. Dehydration reaction because basically you're losing water out of the molecule. All right, yeah.
So what defines which kind of like hydroxyl group will be coming from the sugar or from the phosphate? Because here we see that phosphate is losing hydroxyl group and sugar
Yeah.
is losing just hydrogen.
Yep, you're losing the hydrogen here and then the OH here. So the oxygen from your water is coming from the phosphate group.
But sugar can also lose hydroxyl group and phosphate can also like lose just the hydrogen. But why is it happening the other way?
Why is it happening from the 3' end?
No, like why is the 3' end just losing the hydrogen and phosphate is losing the?
That's a good question. I actually don't know exactly why the oxygen comes off the phosphate. I'm sure it has to do with energetics. I'll have to go ask a biochemist on that one. Good question, though. And actually, there's even a little bit more to the story. It's actually a little bit more complicated than this. And the question I can ask you is, is this anabolic or a catabolic reaction? It's anabolic, isn't it? Okay. Is it endergonic or exergonic? Does it require energy or is this releasing energy? It takes energy to build things, right? It takes energy to build up a complex molecule. If I'm building a protein, if I'm making DNA or RNA, this requires an input of energy. Okay. So as we build our nucleotide, our nucleic acids, DNA and RNA, don't forget we have this directionality that your polymerases can only add to the 3' end. And then when we form this, we have what is called a phosphodiester bond. So a diester bond would typically be something that looks like this. Like that. It's actually an ester bond. A diester bond, of course, would be 2 of these. But because here, this carbon, oxygen, a diester would be carbon right here. But since we've got a phosphate group, there's our phosphate group in there. We have a phosphodiester bond. And this would be the carbon, oxygen, phosphorus, oxygen, carbon. Di just means 2. Ester is like a COC, carbon, oxygen, carbon. This is carbon, oxygen, phosphorus, oxygen, carbon, hence phosphodiester bond. Okay, any questions? Pretty straightforward. This is also called a sugar, because here's your 5-carbon sugar. In this case, it's a ribose sugar. Here's our phosphate. We call this a sugar phosphate backbone. And this is also its primary structure. So the primary structure of a nucleotide is your primary structure of your nucleic acid, DNA and RNA, is your sequence of nucleotides. That's your primary structure. And it's held together by these phosphodiester bonds. And this forms what is called the sugar phosphate backbone. Once again, this is actually really important. There's a couple things happening here. We think about DNA, it's a stable molecule. There's lots of reasons why it's stable. But when you look at the sugar phosphate backbone, the first thing, do you think of it as being hydrophobic or hydrophilic? Can this sugar phosphate backbone form hydrogen bonds with water? Do sugars dissolve in water? Oh yeah, can a phosphate group dissolve in water? So can a sugar phosphate backbone, is it hydrophobic or hydrophilic? It's absolutely hydrophilic, right? It's hydrophilic, and it will clearly form hydrogen bonds with water. That is another really important point about the structure of DNA and RNA in terms of how it functions and how it looks. These are the nitrogenous bases. Interestingly, those molecules are more or less flat, or at least this part of them are more or less flat. And if something's flat, they can stack nicely, can't they? And even though there's some nitrogen and some oxygen in here, they're not quite as hydrophilic as the sugar phosphate backbone. They're less hydrophilic. They're more hydrophobic. Now remember, hydrophilic and hydrophobic is not an either or. There's a range. Some things are incredibly hydrophilic. Some things are very hydrophobic. So like fats, which are almost all carbon and hydrogen, and lots of them are completely, almost always hydrophobic. This is somewhat hydrophobic. It's more hydrophobic than this. This is really hydrophilic. That's important. Okay, any questions? Okay, now, the other thing here, oxygen, nitrogen, and they're attached here to carbons. There's a nitrogen, there's a nitrogen here, and a nitrogen and an oxygen. Are those more electronegative than the carbons and hydrogens that are around them? Yeah. So parts of our nitrogenous bases look like they can form hydrogen bonds, don't they? Yeah. So part of our nitrogenous base is capable of forming hydrogen bonds. Like I said, hydrophobic, hydrophilic is not a total and or. Okay, so I started talking about this. We got our sugar phosphate backbone. This, of course, is an endergonic reaction. Anytime you build something complex, it requires an input of energy. If you're playing with a Lego set, it takes your energy to put it together. If you're building a protein, you need a source of energy. If you're building RNA and DNA, it requires a source of energy. And it turns out that we use what is called, or at least cells use, activated nucleotides. ATP, adenosine triphosphate, is an activated nucleotide. There are 5 activated nucleotides that our cells use. Adenine triphosphate, ATP. There's also guanosine triphosphate, right? You see cytosine triphosphate. So each of our nucleotides comes in an activated form where there are 3 phosphate groups here. Now, another thing about these phosphate groups, why do we call these activated? They have a lot of potential energy. And basically, oxygen's negative, oxygen's negative, oxygen's negative. Like charges repel. As a result of these things repelling each other, these covalent bonds here and here have more and more pressure being put on them. They have more potential energy. So we can use that potential energy to help synthesize our nucleic acid, whether it's DNA or RNA. Okay, are we good? Okay. So anytime we're making proteins or polymerizing complex molecules, it requires energy. And this is just showing how much energy it is. It requires 10.9 kilocalories a mole, basically ATP, to link a nucleotide to RNA. So you need to have at least that much energy. And this is something you'll learn more in I believe, chapter 8, right after the spring break. Okay, let's make some comparisons between nucleic acids and proteins. They're both made of building blocks, the monomers. Nucleic acids and proteins are both polymers. You both make them through condensation or dehydration reactions. They are both endergonic reactions. They both have a primary structure. In proteins, that primary structure is the sequence of amino acids. In DNA and RNA, the primary structure is the sequence of nucleotides. And you can think of like the fact that DNA and RNA store genetic information. You can think of those nucleotides as letters, and letters come together to form words. Just like we have 26 letters to form all the words of the English language, life uses 4 letters: adenine, thymine, guanine, and cytosine. So the information in our nucleotides, the information in our nucleic acids is stored in that sequence of nucleotides. Is that good? Okay. Wow, a sequence of nucleotides codes for a sequence of amino acids. I'm priming you guys for genetics, if you can't tell, right? It's also just, the genetic code is pretty fascinating. Now, proteins have a primary structure, and that primary structure is formed by hydrogen bonding of its backbone, of its amino acid backbone. Here's a difference between nucleic acids and proteins. They both have secondary structure, okay? However, the secondary structure in our nucleotides, Our secondary structure in nucleic acids is formed by base pairing between these nitrogenous bases. And the way that base pairing works is that adenine will form 2 hydrogen bonds with thymine, and guanine will form 3 hydrogen bonds with cytosine. These dots just represent a hydrogen bond rather than a covalent bond. That's why I drew dots rather than lines, because a line would represent a covalent bond. Now you might ask, why not have a covalent bond? Why have hydrogen bonds? Anybody know why, or potentially why? Yeah.
The hydrogen bonds can be broken down using an enzyme, so it allows RNA to copy that DNA easier than a covalent bond.
Yeah, that's right. It's a much easier bond to break. So when you have an RNA or a DNA polymerase, it's very easy for these things to separate out. Enzymes will also break covalent bonds as well. But you're right. If we covalently bonded this, it would be a strong bond, which would hold these things together very tightly, versus a hydrogen bond allows DNA to be separated out much more easily. It allows the 2 strands to be separated equally or easily. Interestingly, what's kind of cool is that if you notice, adenine and thymine form 2 H bonds. Not bombs, bonds. And then this one has 3 H bonds. Interestingly, have you guys ever heard of something called thermophilic bacteria? Or thermo means heat, right? Phile means loving. There's these thermophilic bacteria in archaeans, prokaryotes, very simple single-celled organisms that can live in like water 200 degrees. 200 degrees, right? And interestingly, their DNA contains a lot more guanine and cytosine than adenine and thymine, because in the heat, there's a lot more thermal energy that would cause these things to separate out. But by having a higher GC content in their DNA, it helps hold that DNA together more strongly. So there's actually an evolution toward a higher GC content in these thermophilic bacteria. Just kind of fascinating how that works out. Okay, so here it is. Here's our secondary structure. And DNA and RNA is formed by hydrogen bonding between the base pairs. Adenine always pairs with thymine with 2 hydrogen bonds. Guanine always pairs with cytosine with 3 hydrogen bonds. So, one thing is, if you're going to live in a hot environment, it's better to have more GC, more guanine and cytosine. Yeah?
Why does adenine and thymine not create the 3rd hydrogen bond compared to guanine and cytosine?
Well, when you line them up, so first of all, I think that's a really good question. Basically, these 2 strands are running anti-parallel to each other. So one's going east, the other one's going west, if you're like on I-40, right? So this would be my 5' carbon here, this would be my 3' carbon here. And it orients our nitrogenous bases so that here on the C and G, that you can get the 3 hydrogen bonds here. Now, if you look at this one, even though there's an oxygen right here, this could form a hydrogen bond, but across from it is a carbon and a hydrogen. Equal electronegativities, or roughly similar. They're not quite exact, but similar electronegativities. So this can't form a hydrogen bond with that because of the orientation of the way these things face each other. Do you notice any other similarities here, or any other patterns? Not similarities, but any other patterns? Adenine and thymine, guanine and cytosine. Purines, pyrimidines. You notice something else? Pyrimidine, purine. Pyrimidine, purine. A and G, are these purines or pyrimidines? And of course, purine always pairs with a pyrimidine. Okay, that does something else here. So there's a lot going on with our DNA molecule. So basically, we have our primary structure, which is our sequence of nucleotides. We have the primary structure as a sugar phosphate backbone, which is very hydrophilic. It is forming hydrogen bonds with water. The nitrogenous bases are less hydrophilic than the sugar phosphate backbone. But these nitrogenous bases are not totally hydrophilic. They can form hydrogen bonds too, and they form hydrogen bonds with themselves. A purine with a pyrimidine, A with T, G with C. So this is forming our secondary structure. These molecules are also flat, so they tend to stack very nicely, forming these hydrogen bonds between them. And that, because this is hydrophilic, it basically turns these away from the water, the outside, and into the inside, and further stabilizes the molecule. Good? Okay, and of course, one thing I forgot to mention is that the sugar phosphate backbone, of course, is held together by strong covalent bonds, whereas this is held together by weak hydrogen bonds, which allows your strands to be pulled apart, which allows the secondary structure of your nucleic acids to be easily pulled apart. Okay? All right. Now, of course, this is DNA structure and function. So DNA is double-stranded. Here's one strand, here's the other strand, and of course they're anti-parallel to each other. One is going in the 5' to 3', the other one is going in the opposite direction. Okay. And this Chargaff's rule that I was telling you about is that in any DNA, when they were discovering what DNA was and what it looked like, Chargaff in I think the 1940s realized that in any strand of DNA, the number of purines always equaled the number of pyrimidines. And at the time, it was originally thought that DNA was such a simple molecule that it was just scaffolding for the proteins that did all the work, that stored all the information. Okay, there's our secondary structure of DNA, the base pairing. You can see, of course, your phosphodiester bonds here. Here's your pyrimidine, your purines, forming your hydrogen bonds here. So on a test, I would normally give you something like this and say label everything. Which one are your purines, which ones are the pyrimidines, which nitrogenous bases are the adenine, thymine, guanine, and cytosine. Okay, any questions? Okay. Now, molecules also have a tertiary structure as well. And in proteins, the tertiary structure of a protein is really, really varied. And it's determined in large part by interactions of your side chains of your amino acids. Okay? And one of the things that gives proteins their incredible functionality, like why they can do so many things, is because, well, quite frankly, they can form many, many different types of shapes based on these different side chains they have. So the tertiary shape is often considered to be the three-dimensional shape of a molecule, whether it's a protein or DNA. Now, honestly, when I read the book and I was just making sure I had everything straightened out in here, it called the double helical structure of DNA secondary structure. Now, I was a little taken aback by that because to me the secondary structure was a base pairing amongst the nitrogenous bases, right? So when I talk about base pairs, that's an A with a T. So I looked it up to the Wikipedia really quick and consulted a few other textbooks just to make sure I hadn't forgot or lost or got something wrong. And I'm right. Tertiary structure is your overall three-dimensional structure of your molecule. So what happens is your DNA forms basically a twisted ladder. Okay? You guys all seen ladders, right? The outside of the ladder that holds it all together. And our DNA and RNA, or our DNA, that would be a sugar phosphate backbone. And the rungs, of course, are the nitrogenous bases in the middle. And then what happens is, imagine taking that ladder and twisting it into a helical shape, hence double helix. That three-dimensional shape is also its tertiary structure. So I think I'm going to update this here. And the book said that the tertiary structure with these supercoils and things like that is it wraps around histones and other proteins that help organize the DNA inside the nucleus. I think that's actually a quaternary structure, but yeah. Tertiary structure, to keep everything simple on the same plane here, tertiary structure of DNA would be its three-dimensional shape. And it's twisted into this double helix. So you've got these sugar phosphate backbone twisting around like this, and this is attracting all the water. The more hydrophobic, flattened purines are in the middle, keeps the water out. They can interact with each other through van der Waals forces up and down the helical structure of it, which further stabilizes this molecule. And of course, with the DNA, you're using also the deoxyribose sugar here, which is more stable. And your thymines, so they won't switch to cytosine, representing a mutation. Are we good?
How does it twist? Why does it twist?
That's a really good question. So much like a protein will often fold into its correct shape, the reason why it does that is through interactions with both the nitrogenous bases and interactions with the backbone here and the interactions with water. And this is like one of at least 3 different shapes it takes, that DNA can take. There are other shapes that it can form, but this is the most stable in water based on interactions of the sugar phosphate backbone and the nitrogenous bases. So it's an energetically favorable state for it to be in. So if it's energetically favorable, it's also stable. Okay, good questions. Anybody else? Yeah.
So depending on the sequence of the nucleic acids, could it be more twisted or less twisted?
No, it's always going to have that's a really good question. And it's always going to have the same twisting because your number of purines always equals the number of pyrimidines. So even if I have a high GC content, I still have the same number of purines and pyrimidines. And in fact, your question is really insightful. There are these grooves, and they're called the major and the minor grooves. And the size of those grooves is very important for the DNA and RNA polymerase, especially DNA polymerase, to get into those grooves, help pull the DNA apart, and then replicate it. And when things like, you ever heard of benzene? Benzene is this really nasty thing. Sorry, I'm spoiling some genetics for you, but how can you not with this, talking about DNA and RNA? Benzene, of course, you know, is a hexagonal flat ring. You guys have had organic, right? A little bit? No? Here. There's our benzene ring. It's a flattened ring. What happens with benzene is it slips in here like this, and it changes the size of these grooves, the major and the minor grooves, and it causes the DNA polymerase to not replicate DNA accurately, introduces mutations. That's why benzene is a carcinogen. That's why benzene causes cancer, or the cancer-causing agent, I should say, because it actually changes the twisting shape of it. Good questions. Anybody else? Okay. I already talked about that. Okay, very quickly. In this class, we're not going to really get into the nuts and bolts of DNA replication. Like I said, that's more for genetics. I find it just absolutely fascinating, like the number of proteins involved with replicating DNA. But for us today, all I'm going to say is that basically the replication is what we call semi-conservative. And what happens is you take your DNA, it's a double helix, and the reason why it's got those hydrogen bonds holding the 2 strands together, kinds of proteins involved with separating out the DNA. And then I've got, is my original parent strand, then you separate it out, and then each parent strand serves as a template for a daughter strand, and semi-conservative, which is different than like a conservative replication, where you would have these two daughter strands would be out here by themselves. But whenever you replicate DNA, each new strand will have part of the parent and part of the daughter. And all of this directionality, like I said, really, really matters for DNA synthesis because you can only add nucleotides to the 3' end, which means that there are some weird things happening on the other strands. Okay, let's talk about RNA structure and function. So we know that DNA stores genetic information. It's a fairly stable molecule. Uses a deoxyribose sugar. It uses thymine because thymine's more stable, and it forms a double helix, which once again is an energetically favorable place for that to be. Which if you're going to store information for the long term, be stable, right? And the other thing is basically DNA stores genetic information. That's it. That's like all it does. RNA is a bit different. There's a lot of similarities here, other than the fact we know they use the ribose sugar, they use uracil because it's cheaper. But one of the other key differences, DNA is double-stranded, RNA is single-stranded. And that should remind you of something. The proteins are single-stranded. Now, of course, large protein complexes have quaternary structure with 2 or more polypeptides coming together to form a protein. But a lot of proteins have different shapes, and all they have is primary, secondary, tertiary structure. But they're single-stranded. RNA is a versatile molecule. It is also single-stranded. And it has a primary structure of your sequence of amino acids, your sequence of nucleotides. The secondary structure is formed by base pairing. Okay, and notice helical structure here as well. But this is one strand. It's all one strand. So RNA is single-stranded. Once again, that's incredibly important because RNA is a very versatile molecule. And if you're going to be versatile, you got to have different shapes, right? Yeah, if you're in different shapes, you can do different things. So by being single-stranded, this can actually form different shapes. Okay. Yeah, so we've talked a lot about the RNA, the DNA, the differences here, but it's a little bit more reactive. It's single-stranded and it forms different shapes. For example, there's these things called tRNAs. RNA is versatile. We already know that it can store genetic information. I mean, it's made up of nucleotides, right? So it can carry genetic information, but it does other things as well. Because it can form multiple shapes, like I said, it has different structures, it has different things it can do. For example, this is a tRNA. tRNA stands for transfer RNA. So you know that proteins are made inside the ribosomes, right? Well, ribosomes are these large molecular complexes that are made up of ribosomal RNA plus proteins. And these things called tRNAs. This, of course, is just showing kind of the secondary structure of it, not showing the full three-dimensional structure of it. What tRNAs do is they bring amino acids into the ribosome and line up the specific amino acid with what's called a codon. So your messenger RNA, you're inside of a cell, you got your DNA inside the nucleus. DNA is just storing information, doesn't do anything with protein synthesis. It just has the information to do it. So you copy the information to a messenger RNA. The messenger RNA goes out into the cytoplasm of the cell, binds to this thing called a ribosome. And then these things called a transfer RNA, they are out in the cytoplasm. They attach to specific amino acids. And then they attach to the amino acid. Then they go to a ribosome. The ribosome aligns the transfer RNA based on this codon, which is a sequence of 3 nucleotides. And then brings in the right amino acid at the right time. So you're already starting to see the point. There are 3 types of RNAs doing different things because they have different shapes. This is a tertiary structure of a transfer RNA, I believe. I forget. I think so. Looks like it. So this is right here is what lines up with the codon. You guys know what codons are? Sometimes I get way ahead of myself. A codon is a sequence of 3 nucleotides, like it could be any CCC, UGA, CCC, ATT, whatever. But this sequence of nucleotides code for a specific amino acid. That's the code of life. That's genetics. Isn't that cool? That's your tertiary structure of your RNA that brings amino acids to make proteins. And I'm just comparing it to a large protein complex called cytochrome oxidase. This is found inside your mitochondria. It's part of how your cells make ATP, but they both have these, complex 3-dimensional structures. This is an example of a ribosomal RNA. So your ribosomes are made up of proteins and ribosomal RNA, also known as rRNA. This is where your actual proteins are synthesized in your ribosomes. And interestingly, the ribosomal RNA is held in the right position by proteins. So ribosome is this large molecular complex made up of proteins and rRNA. And interestingly, it's the ribosomal RNA that actually stitches the 2 amino acids together. It's catalyzing a reaction. And the proteins help line everything up. And this is what a ribosome actually looks like. They've got a small and a large subunit here. This would be the small subunit, this is the large subunit. And they only come together when they're actually making a protein. Wow, we actually know what these things look like. Isn't that astounding? Like the amount of work that went into that is astounding, and the fact that we can actually figure that out. There's a simplified version of a tRNA, brings amino acids to the ribosome. And then you have messenger RNA. Messenger RNA, of course, just carries genetic information from the DNA to the ribosome. So we're starting to see RNA is pretty versatile, right? Okay. So in most classes, you'll learn about RNA, ribosomal RNAs, transfer RNAs, messenger RNAs. These are RNA, tRNA, mRNA. We've known about them for a long time. Now here's where it gets really fun. Now just kind of sit back and relax for a few minutes. I'm not going to test you on anything here. And if I did, you could just look it up. But this is what's really interesting. RNA, like I keep hammering home, it's just this really versatile molecule. It can, it can carry genetic information. It can facilitate its own reaction as well. But more than that, it turns out that RNAs have incredible number of functions inside of our cells that we're only just now learning about. So, in the 1990s, we sequenced the human genome, and that was like enormous in terms of like our understanding of what all of that DNA in our cells are doing. We had to redefine what a gene is. We thought that genes just code for proteins, right? Well, actually, they also code for lots of different RNAs that regulate the activities of the cell. They regulate, like, when do you turn a protein on? When do you turn it off? How fast do you degrade your protein? How fast do you make your RNA last? I mean, it's just doing all kinds of things. So another thing they learned, this is just for fun, when they started sequencing the human genome in the early 90s, they were taking out bets, like how big is the human genome going to be, right? And they're like, well, there's going to be about 100,000 genes. There's more like 20. And they're like, how in the world do you have over about 100,000 proteins in your body with 20,000 genes. And it turns out that genes, you can have a gene to make a protein, but embedded in that gene could be these commercials called introns. You can slice all that up and re-put together the genes in different orders, and you can have one gene actually make like 2, 3, 4, 5, 10, 20 different proteins from a single gene. It's called the introns and exons. Exons actually code for the proteins, and then the introns are the intervening units. But you can take your exons and take a gene with all these exons and introns. Introns are like commercials. Splice out the introns, take your exons, and re-put them together in different patterns and get different proteins. And this is an RNA involved with that called a spliceosome. So it can take you'll take your gene, you'll copy it in the messenger RNA. This RNA here called a spliceosome, snRNA, will cut that messenger RNA up and restitch it back together to make a new protein. It's just, how does your cells know how to do that? It's just crazy. MicroRNAs, so you can have RNA go out into your cell. Wait, we don't need that protein anymore. Let's silence it for a few minutes. So there are ways that these microRNAs will silence an mRNA. So you can regulate protein synthesis really fast based on the cell's needs. All right. And of course, not only are they used in regulation, as I said, these things can actually facilitate their own chemical reaction, which on Thursday is going to be very interesting for origins of life. People thought, wow, here's a molecule that can store information and catalyze reactions. What more could you want? Well, a better explanation for one. Okay. Yeah. The role of RNA inside of our cells is been a hotbed of discoveries. Like approximately 30 scientists have won Nobel Prizes studying RNA. Isn't that amazing? Like not proteins, but RNA and all of the different functions. So what I was trying to get at is that there's just a lot. We still don't know everything. Okay. And lastly, you guys want to hear a little controversy about the discovery of DNA? The books don't talk about it so much, but Rosalind Franklin has become much more popular today than in the past. But basically, Watson, Crick, and Wilkinson won the Nobel Prize for Physiology, Chemistry, or Medicine, one of those in like 1962 for their discovery of DNA. Well, it turns out that, there's their Nobel Prize. Oh, I didn't get the picture in there. The way that you used to determine the structure of a molecule was through X-ray crystallography. And she happened to be one of the world experts, one of the world's best people at X-ray crystallography. And she took the image called Photo 51 that was famous and took that image. And these guys were working on the structure of DNA. She was working on the structure of DNA, her and her students. And so she left King's College, where she couldn't even go to lunch in the lunchroom because she was a woman in the 1950s. So she couldn't go have lunch with her male colleagues. God, man, I'm sorry. I get angry about that. Like, what world does that work? At any rate, she left King's College to go to another college that was much more conducive to women in science. And her other person there called Maurice Wilkins gave the photo 51 to these guys who looked at it, interpreted it, and immediately published on the structure of DNA. And then they went on to win the Nobel Prize with the guy that gave them the photograph and gave Rosalind Franklin an acknowledgement. Unfortunately, she did not get a Nobel Prize for several reasons. The main one is she died in the late 50s of ovarian cancer, probably because she kept walking in front of the X-ray beams all the time. And those beams were pretty powerful back in the 50s. And that was really sad that she actually, she was 35 when she died, basically. But She basically discovered the double helical structure of DNA. But then she also went to go work on the tobacco mosaic virus. She figured out like the structure of the virus. And her postdoc that was working on it with her went on to win a Nobel Prize in like 1980. And he got up and said that this work would not have been possible without the founding what she did on the work of the tobacco mosaic virus. So think about this. By the time she was 35, she had done the work of 2 Nobel Prizes. There's only other like one woman in the world that ever won 2, and that was Madame Curie, right? I mean, she basically did that too. Pretty cool. I think they even made a probe named after her now. So that's cool. All right, so on Thursday, we're going to talk about the RNA world hypothesis and other theories of abiogenesis. And just Google it. See what you guys think. I'm curious as to what you guys think.